about the male reproductive system, and we were talking about the spermatogenesis. Okay. Um, Last time we were talking about the spermatogenesis. We talked about the meiosis, the differentiation of the germ cells, the meiosis, and we said that at the end of the spermatogenesis, we have the spermiogenesis, which is the cytodifferentiation of the spermatids. And following this differentiation or cytodifferentiation of the spermatids, the spermatids will become spermatozoa. And we said that so we have many events that will occur to the spermatid and that will lead that it will become a spermatozoa. And now in this session we are going to talk in details about this spermiogenesis. And we said also that at the end of the spermiogenesis, the produced spermatozoa will be released in the lumen of the seminiferous tubules, and this release of the spermatozoa in the lumen is called the spermiation. This process is called the spermiation. Okay. Now, what happens during the spermiogenesis? So this phase where we have the transformation of a spermatid to a spermatozoon. So as we said, the spermiogenesis is the process by which the spermatids, which are the gametes produced by the meiotic division, they are transformed into motile mature spermatozoa. Now, first of all, we will have as you can see, we will have a variation in the spermatid's shape during the cytodifferentiation. They will become more elongated. They will have a flagellum or a tail, okay, that was not present before. Their nucleus is going to be more elongated and more compact, and the cytoplasm will be reduced. So there is many changes. First of all, we will have the formation of the acrosome, which is formed from the Golgi apparatus. So as you can see, this is a spermatid, an immature spermatid. An immature spermatid, yes. This is a spermatid formed after the meiosis. And this is a cell containing mitochondria, a Golgi apparatus, centrioles, a nucleus, etc. And so the Golgi apparatus of this spermatid will produce vesicles, okay, that will aggregate to form the acrosome, what we call the acrosome. And the acrosome is this dark purple structure on the top of the nucleus. Okay, so the acrosome will be formed from vesicles of the Golgi apparatus, and this acrosome in fact will contain enzymes necessary for the fertilization. The enzymes in the acrosomes are necessary or are essential for the spermatozoon to cross the membrane and the zona pellucida of the oocyte in order to fertilize or in order to fuse, to have a fusion of the gametes. So we will have the formation of the acrosome from the Golgi apparatus. We'll have the centrioles that were in the cytoplasm. Those centrioles will migrate to the posterior side of the cell. When I say anterior and posterior, in fact, the anterior side is where we have the acrosome, and and this side is going to be the head, the anterior side of the head of the spermatozoon. And when we say posterior, we mean at the opposite side of the acrosome, where we are going to have the flagellum. So those centriole will migrate to the posterior side, opposite to the acrosome of the cell. Also, the mitochondria will migrate to this posterior side, as you are going to see here. And the cytoplasm also will migrate to this posterior side. So we will have also a reorganization and maturation of the nucleus. We can see how the shape became different. And also the cytoplasm, we said it will migrate here, and then the excess of the cytoplasm will be removed. We are going to talk about those changes in details. Now, as I said, the Golgi apparatus will elaborate membrane-bound vesicle called the acrosomal vesicle, and this acrosomal vesicle contains carbohydrates and hydrolytic enzymes, and it will be applied to the upper side of the nucleus, on the anterior side of the nucleus or of the cell. Okay. And then it will be here, and then it will elongate on both sides of the nucleus, and it will cover almost 2/3 of the size of the nucleus. Okay. And meanwhile, at the same time, we will have the centrioles that will migrate to the posterior side. We will have to, and those centrioles will form a T shape. Okay, as you can see, one is parallel to the axis of the flagellum, this one here, and the other one is quite perpendicular to it, so they are forming a T shape. Those centrioles here, which are parallel to the axis of the flagellum, this centriole is the distal centriole, is called the distal centriole. And in fact, this distal centriole will be at the origin of the flagellum, will form the microtubules, the central microtubules of the flagellum. So the centriole aligned parallel to the long axis of the nucleus is called the distal centriole, and will elongate to form a flagellum. In fact, this centriole will form the central microtubules of the flagellum, or more specifically of the axoneme of the flagellum. The other centriole is called the proximal centriole, and we will talk about its role. Now, once this flagellum is elongating, when it is elongating, we have also other elements that will come and will surround this central element, this axoneme of the flagellum. We have other elements like what we call the coarse fibers or the outer dense fibers. So we have 9 outer dense fibers or 9 coarse fibers that will come and surround also this axoneme, but not all along of it. We will see also where. We'll have the mitochondria also that will migrate and will sit in this region, in the upper region of the flagellum. And also the cytoplasm that will migrate and will be mostly here in this upper region of the flagellum also. And then the excess of the cytoplasm, which will be called the residual body, will be removed, and the removed parts will be phagocytosed by the Sertoli cells. Now, before that I go further in the details of this spermiogenesis and what happens more in details, it's better to, I would like to explain or to have an idea about the structure of the spermatozoon, to understand its structure. Then we will we will continue the explanation about the spermiogenesis.
دكتورة
So the spermatozoon, yes, do you have a question?
no please فيك تحطي البوينتر انتوا عم تشرحي مش الماوس
نعم.
البوينتر can you use the pointer the
You are
representation
not seeing the mouse?
انه اذا فيك تحطي البوينتر احسن لانه ببين اكثر
Okay, okay. Now, the spermatozoon is composed from 3 regions: the head, the neck, and the tail. The tail is the flagellum, in fact. So we have the head, the neck, and the flagellum. The head contains the nucleus and the acrosome. Okay, and the nucleus will contain a highly condensed chromatin. We will see now that the DNA or the chromatin inside of the nucleus of the spermatozoon will get very condensed. Okay, and we will have also the acrosome that we said it contains the proteolytic enzymes responsible for the penetration through the zona pellucida of the oocyte. The neck will contain a connective part that will connect the tail to the head and will allow their harmonious movement. And also it will contain the proximal centriole, this proximal centriole here that we have seen here, okay? And this centriole will, in fact, will initiate, after the fertilization, the centriole will initiate the first division. It will initiate the formation of the spindle in the zygote during the first division of the zygote. Okay, so this centriole is important for the later divisions, for the first division of the zygote after the fertilization. So this is regarding the neck. It contains a connective part which attaches the head and the flagellum and the proximal centriole, and we have also the tail or the flagellum. Now the flagellum is composed from three parts. We have this upper part of the flagellum is called the midpiece. Okay, then this is the largest part. This large part is called the midpiece, and then we have the principal piece, which is the piece directly under the midpiece, and at the end we have the end piece of the flagellum. Now, all along the flagellum, we have one component that is present all along of this flagellum. This is the axoneme. Okay, the axoneme is present at the center of the flagellum. Okay, and it is here you have the structure of the axoneme. You don't have to know it, but you can see that at the center of the axoneme we have the pairs of microtubules which were derived from the distal centriole, and we have also 9 other pairs or doublets of microtubules. So this axoneme is at the center of the flagellum. Now, in the midpiece, and then this axoneme will be surrounded by different elements according where we are, according to the part of the flagellum. For example, in the midpiece, this axoneme will be surrounded by 9 bundles of outer dense fibers. So we will have 9 outer dense fibers which will come and surround the axoneme. And in fact, those outer dense fibers, they protect the spermatozoa from shear forces during their transport in the genital tract, and also they allow certain flexibility for this flagellum. Okay, so those outer dense fibers will come and surround the axoneme in the midpiece, and around them will be situated the mitochondria. Okay, so we will have the mitochondria also, or that will form a sort of mitochondrial shed. And as you know, the mitochondria, they produce energy by hydrolyzing the ATP. So the energy produced here by the mitochondria will be essential for the movement of the flagellum and for the movement of the spermatozoa. So from outside to the inside, in the midpiece, we will have the mitochondria, and then the outer dense fibers, and then the axoneme. We can see here a cross section of this midpiece, where we have at the center the axoneme. This is the axoneme at the center. Those in black, they are the 9 bundles of outer dense fibers. Each one is a bundle of outer dense fibers, and here you have the mitochondria. Now, in the midpiece, we have the central element is the axoneme, And around the in the principal piece, excuse me, we have the axonium in the middle, and around this axonium we have also the outer dense fibers, the ODF, 9 bundles of ODF. But this time, um, surrounding the ODF, we will not have mitochondria, but we will have a fibrous sheath. Okay, so we will have a sheath of fibers that will surround those ODF in the principal piece. And in fact, this fibrous sheath serves as a scaffold for different proteins necessary during the motility of the sperm, that will regulate the motility and the maturation of the sperm and its capacitation that we will see later. Okay, so here is a cross section of the mid piece. We will have the axonium in the middle. We have those outer dense fibers surrounding the axonium, and then we have this fibrous sheath surrounding the outer dense fibers. So in the principal piece, from the outside to the inside, we will have the fibrous sheath, 9 ODF, and then the axonium at the center. And at the end, we have the end piece of the flagellum. And in this end piece, we only have an axonium. We don't have outer dense fibers, neither mitochondria. Okay, so we only have the axonium present in the end piece of the flagellum. Here you can have a cross section of the end piece. In this cross section, look, all of this is the axonium. Okay, this is the axonium in the middle. Here you have the principal piece, you have the axonium here, those are the ODF, and this is the fibrous sheath. And in the mid piece of the flagellum, we have the axonium in the middle, the 9 ODF here, and then the mitochondria surrounding the all. Okay, this is regarding the structure of the spermatozoon. Now we will go back to talk about the spermiogenesis. So as I said
دكتورة
before, yes.
بس سؤال، الأشياء اللي كنت عم تقوليهم قبل كانوا introduction ولا إنه كنت مبلشة؟ لأن حسيت يعني مطيشرين بكذا فقرة هن، فكنت عم تعملي introduction؟
For the spermiogenesis?
Yes.
Yes, يعني I was going from the general to the more detail. يعني I was just talking about general features to go further in details later.
اوكي، يعني هلا حنبلش فيهم.
هلا حنشوف الديتيلز اكتر يعني، بس بالاول حتعطيكم فكره عامه انه حتى تفهموا احسن الجنرال ايديا.
اوكي تمام.
So we talked about the acrosome formation, and we said that this acrosome in fact is composed from small vesicles coming from the Golgi apparatus, and those small vesicles will gather together and fuse to form this large vesicle called the proacrosomal vesicle. This proacrosomal vesicle, which is first formed here, this proacrosomal vesicle, okay, will migrate and will sit on the top of the nucleus to become the acrosome. So it will be placed at the future anterior pole of the spermatozoid nucleus, of the spermatozoon nucleus, opposite to the flagellum, and will spread until covering about the 2/3 of the anterior of the nucleus. Okay, so this acrosome here, it will spread to cover the 2/3 of the anterior part of the nucleus. And as we explained, it contains hydrolytic enzymes that will hydrolyze the zona pellucida of the oocytes. And those enzymes, they are like the acrosin and the hyaluronidase, okay, essential for the fertilization and for the penetration during the fertilization. And those enzymes in the acrosome will be released at the time of the acrosomal reaction and will be necessary for crossing the zona pellucida during the fertilization. Now, what happened to the nucleus? We said that this nucleus will undergo many morphological changes and other important changes in the DNA. In fact, first of all, we had a round spermatid having a ovoid central nucleus, as you can see. This is a round spermatid with an ovoid nucleus, and later this nucleus start to begin more elongated, will elongate in shape and will be smaller in size also. So and this ovoid central nucleus in the spermatid contained dispersed chromatin, okay, a dispersed, not condensed chromatin, and this nucleus has 1 or 2 nucleoli. Now we start having an elongation phenomenon of the nucleus, and which goes along with a condensation of the chromatin. So we will have a nuclear elongation and a condensation of the chromatin. In fact, what happens to this chromatin? We know that we have nucleoproteins attached to the DNA or in contact with the DNA, which are the histones. Now, the histones or those nucleoproteins are going to be modified. So they are going to be replaced by other nucleoprotein which will be more basic. And this modification of the histones will be made in 2 steps. In the first step, we will have a partial replacement of the histone, and they will be replaced by, so the histones are basic, but they will be replaced by other basic transition proteins, okay? And those transition proteins, since they are basic, they are rich in arginine, histidine, and lysine, and they contain also cysteine. And then those transition proteins, in the second step, they will be removed and replaced by protamines. So at the end stage, the histones will be replaced by the protamines, and those protamines, in fact, they are more basic than the transition proteins. They are also rich in arginine and cysteine. And in fact, those protamines, they are the responsible factor of the compaction of the DNA or of the chromatin during the maturation of the spermatids. So what makes this DNA condense is the presence of those protamine, and more particularly the cysteine groups or amino acids of those protamines. We will see how now. Okay. So those protamines, they are basic nucleoproteins. They are synthesized in the cytoplasm and then transported to the nucleus. So once they are synthesized, they get phosphorylated, and during their they are phosphorylated during their transport to the nucleus. They reach the nucleus and they get bound. They get to bind the DNA. Okay, and once they bind the DNA, they will be dephosphorylated. Following this dephosphorylation, we can have some interaction between the sulfur groups of the cysteine amino acids. I don't know if you know the structure of the cysteine, but you have an SH sulfur groups in the cysteine amino acids. Now, those sulfur, those SH groups on each cysteine amino acid can form a bond with another SH group of another cysteine on another protamine. So we will have the formation of disulfide bridges, of bridges between 2 sulfur groups, belonging to different cysteine amino acids. And those bonds of cysteine that will be formed between all the protamines and different protamines, you know that the protamines are surrounding the DNA also. And since we are forming this bonds of cysteine, of disulfide, it's like we are forming a net, a net around the DNA. So that's how, by forming bridges, the disulfide bridges, we will have a compaction of the DNA. Okay. So this DNA will become imprisoned inside those inside those protamine and inside this network of disulfide bridges, and it will become more compacted. And now this compaction characterizes the nucleus of the mature spermatozoa. So we reach this degree of compaction in the mature spermatozoa, not in the immature spermatids. We have this degree of compaction in the mature spermatozoa, and in fact those protamines are nucleoproteins that we will find only in the spermatozoa also. We don't find them in other type of cells, and the compaction of DNA or chromatin in other type of cells do not occur due to the presence of protamine. So this is a mechanism specific to the spermatozoa. So here we had modification of the chromatin composition. We replaced the histidine by other more basic nucleoproteins, which are the protamines, by passing through a step where we have first the replacement of those histone by transition proteins, and then the transition proteins are replaced by the protamines. And so this compaction due to the compaction of chromatin, the nucleus can be reduced in size, and it can have a smaller size, since the chromatin is not anymore spread in a large volume. So the chromosomes will become closely packed in small volume, and the shape of the nucleus can become elongated and narrow. And also this compaction of chromatin ensures the protection of the sperm genome. And since the DNA is very compacted and when it is compacted, it will not be accessible for proteins or transcription factors and other factors in the nucleus. So this compaction will ensure its protection, and it will protect it from any mutation, any kind of thing that can happen to this DNA. So this compaction will ensure also the protection of the sperm genome. And at the end of the nuclear reorganization, the nuclear protein or the nucleoproteins in the nucleus of the sperm cells will be composed at 80% from protamines and 20% from histones. So at the end, the nucleoproteins surrounding the DNA in the sperm cells will be mostly composed from protamine, 80%, and we will still have 20% of histones as nucleoproteins. Now, what happens to the flagellum? During the flagellum formation, as you know, at the beginning we had a round-shaped spermatid where the centrioles are present in the cytoplasm. We have the mitochondria, okay, and we have a chromatoid body in the spermatids. Now, for the flagellum formation, we need, in fact, the centrioles are involved in this in the formation of this flagellum, and also the chromatoid body. We will see what is this chromatoid body. Now, as I said earlier, that the centrioles, it is mostly the distal centriole, this one parallel to the axis of the flagellum, that will form the doublets of microtubule of the axonium, and that's how it will be and it will start forming the flagellum. So the centrioles will migrate to position themselves at the future posterior pole of the nucleus, opposite to the acrosomal vesicle or to the acrosome. And then the proximal and distal centrioles have a T configuration. Okay, they will have a T configuration. The proximal centriole will initiate the first division of the zygote, as we explained earlier, and the distal centriole will give rise to the doublets of microtubules of the flagellum, which are the central microtubules of the axonium. And then in the neck region here, we have also what we call the segmented colons. Those segmented colons will be the elements where the dense fibers start to form. So they are the starting point of the dense fibers. So here you have the neck region, we have the centriole, the proximal centriole, we had and we have also here the segmented colons, and here in the flagellum, in the midpiece of the flagellum, we have the outer dense fibers, and as you can see from the segmented colons, we will have they constitute the starting point of the formation of the outer dense fibers. And at the same time also the mitochondria, they will migrate and will gather at the middle part of the flagellum to form a mitochondrial sleeve or a mitochondrial sheath around the dense fibers. Now also in the flagellum, we said that we have the formation of the doublet of microtubules from the distal centriole, then we have the outer dense fibers. Then also we have, we said we have those mitochondria that will come and they will get positioned here in the midpiece. But also we will have a structure called the annulus, which is a sort of constriction here between the midpiece and the principal piece. And this is a circular structure that will be positioned here. And this circular structure is called the annulus. It is present at the end of the midpiece, and it will determine the boundary between the midpiece and the principal piece. And this annulus will prevent the leaking of the mitochondria from the midpiece to the principal piece. So it's like a constriction point here, okay, to prevent the leaking of the mitochondria to the other parts of the flagellum. And this annulus here is composed from what we call the chromatoid body. And the chromatoid body, in fact, is a ribonucleoprotein granule. So it is a granule, a complex of RNA and RNA binding proteins. That's why we call it a ribonucleoprotein. It's composed from RNA and RNA binding proteins. Okay, so the annulus is composed from this chromatoid body. Now, regarding the reorganization of the cytoplasm, as we said, we have the cytoplasm that will come and will slide to the posterior side of the nucleus, and it will slide to the midpiece, to the region of the neck and to the midpiece of the flagellum. And the excess of the cytoplasm is called the residual body. This excess of cytoplasm or residual body will be removed during the spermiogenesis. So the differentiating spermatids, they get rid of it, and this residual body will be phagocytosed by the Sertoli cells. And now we will obtain this sperm cell at the end. As you can notice, we have eliminated the residual body, but we still have small excess of cytoplasm here. This small excess is called the cytoplasmic droplet that will be kept in the spermatozoa, in the formed spermatozoa. So the mature spermatozoa formed from the spermiogenesis will have the cytoplasmic droplet, and this cytoplasmic droplet will be removed in later stages during the maturation of the spermatozoa in the epididymis. So the spermatozoa will be released in the lumen of the seminiferous tubules with this cytoplasmic droplet that is connected to the midpiece. Okay, and it will only be eliminated in the final maturation of the spermatozoa in the epididymis. Is it clear till now?
Yes, clear.
Okay. Now we are going to talk about the abnormalities that can happen to the spermatozoa. We have some anomalies that can arrive to the shape of the spermatozoa, and here we will talk about the morphological abnormalities, and we have also anomalies that can touch the number and the motility of the spermatozoa. And this we have also the sperm survival. Also, we can have a problem at the level of the survival of the sperm cells. Now, what are the morphological abnormalities that can happen to the spermatozoa? In fact, those anomalies, they can arrive to the head, the midpiece, or the flagellum. Yani we can have anomalies touching each one of those parts. And in fact, when we have an ejaculate of spermatozoa containing multiple morphological abnormalities, we talk about teratospermia. So the teratospermia means that correspond to an ejaculate of spermatozoa with multiple morphological abnormalities. Now, what are the abnormalities that can touch the head region? In the head region, some spermatozoa, they can have a big head. So here we can talk about a macrocephalic sperm cells. We can have a microcephalic head. I don't know if we have a figure here. This the pinhead. Okay, this is a small spermatozoa with a spermatozoon with a small head. Okay, this is a microcephalic head. We can have acephalic spermatozoon or spermatozoa, which means they are spermatozoa without a head. They don't have a head, or we can have an elongated head. I don't know. Maybe there is one figure here. Okay, we can have an elongated head or a piriform head, yani which the shape is like a pear. So here, look at here. Here you have the macrocephalic head. The giant head is a spermatozoon with a large head region. The microcephalic head. You can have okay, this is a tapered head is with an elongated, a narrow and elongated head. You can have a duplicated head, so a head with or a spermatozoon with two heads like this one. Okay, so in this duplicated head spermatozoon, you have two heads, and okay, we have two midpieces or one midpiece. Here is only one midpiece represented, but we can have two midpieces, and the one remaining, the rest of the flagellum is one. Or we can have a flagellum with a round head. Now, regarding the midpiece anomalies, Sometimes the cytoplasmic residues can be still attached to the midpiece, and so the residual body is not detached from the spermatozoa, and it can still attach to the head, but it happens less often. Okay, and we can have a bent neck also, a bent neck, which means that the neck is forming an angle with the long axis of the head. It's like we have an angle between the neck region and the head. And we have the anomalies of the flagella. It can be too short, too long. It can be absent, like this one, in this one. Okay. It can be duplicate, so we can have two flagella for one head, with one head like this one. It can be coiled at its end. Okay, it can be very coiled, or coiled under the head. Sometimes the coils are directly under the head. Okay, and it can be bent, forming an angle with the head also. So this is regarding the morphological anomalies of the spermatozoa. Now we can have anomalies touching the number and the motility of the spermatozoa. Regarding the number anomalies, normally we should have a normal concentration of spermatozoa. In one ejaculate, we should have between 20 and 200 million spermatozoa per milliliter of ejaculate. Now, when we have a low concentration of those spermatozoa in the ejaculate, a concentration lower than 20 million sperm cells per milliliter, we will talk about the oligospermia. So this case is called the oligospermia. And we talk about azoospermia when we have no spermatozoa in the ejaculate. Here we have a problem at the level of the spermatogenesis, and we didn't have sperm cells, so the ejaculate will not contain sperm cells. So this case is called the azoospermia, where there is no spermatozoa in the ejaculate. Normally, so as we said, we should have between 20 and 200 million sperm cells per milliliter of the ejaculate, and 40% in the normal case of those sperm cells should be motile, and 60% should be of a normal size and shape. Now, sometimes we have the percentage of motile sperms that is lower than 40%, and this will lead to motility anomalies. So here the anomalies of the motility are related to the percentage of motile sperms, but also this motility anomalies can be evaluated on the type of movement. Okay, if there is a problem in some sperm cells have a problem in their movement, if we have a problem in the flagella, or if it is very coiled, okay, so this will lead to a problem in the movements. And in both cases, when we have a problem in the movements and in the percentage of the motile sperms, we will talk about motility anomalies. So we can have two reasons for those motility anomalies. And we talk about astenospermia, where the percentage of motile sperms is too low, or their speed is significantly reduced. So when the percentage here of those motile sperm is too low, we say we have an astenospermia. And also is called astenospermia when we have the speed of, or the movement, the speed of those spermatozoa is also reduced. And also we have the sperm viability that can be that can be touched, and it will lead to problems at the level of the fertility and at the level of the number of the sperm cells. So the sperm survival viability is another parameter for assessing the fertilizing ability of the sperm cells. And if we have a poor survivals, this means we are less likely to arrive to fertilization sites. In fact, if the sperm cells, they don't survive for enough long period of time, they cannot survive enough during in the male genital tract, and if they survive enough to be released by the ejaculation, and they should survive for a convenient period of time during the fertilization to reach, to pass from the vagina to the site of fertilization, which is the fallopian tube. So it should survive for enough period of time to reach the site of fertilization. If it will die before, if many of those sperm cells have a problem in their viability and they will die before, this will lead to problems, to fertilization problems and fertility problems.
Doctor,
Yes.
is the sperm would die.
Sorry, I didn't hear you.
What are the reasons that would lead the sperm to die?
I don't know exactly. We can have many cellular reasons. Maybe the apoptosis is triggered. There is a mechanism of the cell death if the apoptosis is triggered. If there is a problem in the seminal, in the nutrients in the medium surrounding those sperm cells, or it can be in expressing some proteins inside of those, in the presence of some factors inside of the sperm cells that will lead to their death.
Okay, doctor, thank you.
Now we have talked about the germ cells of the seminiferous epithelium. The germinal line, cell line of the germinal of the seminiferous tubule epithelium, or of the seminiferous epithelium. Now we will talk about the Sertoli cells, which are the other cellular component of the seminiferous epithelium, and which are the somatic cells of the seminiferous tubules. So we had the germinal cells and the somatic cells. The somatic cells of the seminiferous epithelium, they are the Sertoli cells. So those Sertoli cells, they are large pyramidal cells. You can see them here in gray with their nucleus in green, or here in pink with a purple nucleus. So they are large cells. They they extend from the base, from the basis, or from the basement membrane or the basal lamina of the seminiferous tubule to the lumen. So they are long and they extend all over the seminiferous tubule from the basement membrane to reach the lumen of this seminiferous tubule. So they are also polarized cell. They have a surface lying on the basement membrane, which is the basal surface, and or the basal pole, and they have another pole facing the lumen, which will be called the apical pole. Okay, so that's how they are polarized. And also the distribution now of the proteins inside of those cells will be different. We'll see this in the coming slides. Okay, that's why they are called polarized. So they have two poles, the apical and the apical and the basal pole, and the distribution of the proteins and cytokines, growth factors, will be different between those poles. And they lie on the basal lamina, as we said. They lie here on the basal lamina. The apical ends often extend into the lumen of the seminiferous tubule, and in fact they are large, and they form sort of branches or cytoplasmic extensions that surround and that are in contact with the germ cells. So all the Sertoli cells, they form cytoplasmic extensions that get in contact and enter in contact with the germinal cells, okay, and they can form thin branches to enter and be in contact with all the germinal cells of the seminiferous tubules. So they emit lateral extensions that surround the germ cells at different stages of the spermatogenesis. And in fact, we are going to talk about this. The Sertoli cells, they are the they support the germ cells. They give them nutrition and all the essential elements for their growth and for their proliferation and for the meiosis. All of those elements, they are provided by the Sertoli cells and by the contacts between the Sertoli cells and between the germ cells. And those elements can diffuse from the Sertoli cells to go inside of the germ cells. Now, those Sertoli cells will divide the seminiferous tubules into two compartments. They are going to divide the seminiferous tubules into basal or peripheral compartment here and into an adluminal compartment. Adluminal is for adjacent and luminal for lumen. So they will divide the seminiferous tubules in two parts. One part is close to the periphery of the tube and is called the peripheral compartment or the basal compartment, and one compartment is close to the lumen or adjacent to the lumen and is called the adluminal compartment. Now, how does it come that they will divide the seminiferous tubules into compartments? In fact, the Sertoli cells are connected to each other's and to the sperm cells by sort of junctions, okay? We have the gap junctions, the desmosomes, and the tight junctions that can be present. Now, here you have the gap junctions, the tight junctions and other gap junctions and desmosomes or desmosome-like junctions that are present between the Sertoli cells themselves, and also between the Sertoli and the germ cells. Here in white you have the Sertoli cells and the colored are the germ cells. Now, in the basal compartment, in the peripheral or the basal compartment, we will find the spermatogonia, all the spermatogonial type cells, the spermatogonia, and the primary spermatocytes, which did not enter yet in meiosis. So the preleptotene, what we call the preleptotene spermatocytes, okay? Those cells, we will find them in the basal compartment of the seminiferous tubule. And in the adluminal compartment, we will find the post-meiotic cells. So the cells that entered the meiosis, we will find them in this adluminal compartment. And so in this compartment, we'll find the first cell we will find is the primary spermatocyte that entered the meiosis. Then we will find the secondary spermatocytes, then the spermatids, and then the spermatozoa. Okay, so those Sertoli cells, we said they form junctions between themselves and the germ cells. Now, we have three types of junctions present: the gap junctions, tight junction, and desmosomes. Those three types of junctions, they are present in the peripheral compartment, but in the adluminal compartment, we can find only gap junctions and desmosomes. We don't find tight junctions. And in fact, the tight junctions are junctions between the Sertoli cells. Okay, they are very tight junctions that will not allow the passage of large and big molecule, and they will restrict the they will control those tight junctions, the passage of proteins and molecules from this peripheral compartment to the adluminal compartment. And in fact, it is due to the presence of those tight junctions here between the abdominal, between the peripheral and the abdominal compartments that is responsible for this division of the seminiferous tubules into two compartments. Because due to those tight junctions, we will find elements and proteins here in the peripheral compartment that will not be present, will not be present and are different from those in the abdominal compartment, since not all of the proteins present in this compartment can cross to reach the abdominal compartment due to those tight junctions. So those tight junctions, they are the Sertoli cells, due to the presence of the tight junctions, they divide the seminiferous tubules into two compartments, the peripheral and the abdominal compartment. The exchange between the Sertoli cells and the Sertoli and the germ cells happens through those junctions, but as I said, we have those tight junctions here between the peripheral and the abdominal compartment that restrict, do a big restriction of the proteins and quality of molecules that can cross from this compartment to the other one. And in fact, but if they are like a bridge, so how can the spermatocyte, the primary spermatocyte, we said that before they enter the meiosis, they are present in the peripheral compartment, but once they enter the meiosis, they can pass to the abdominal compartment. Knowing that we have those tight junctions, how those cells can pass from a compartment to the others, to the other. In fact, those tight junctions, when the primary spermatocyte will enter the meiosis, those tight junctions will be disrupted. There is enzymes produced by the Sertoli cells as the plasminogen and the plasminogen activator that will be released by the Sertoli cells, and that will disrupt those tight junctions, so they will be disrupted. In this way, the cell that entered the meiosis can pass from this compartment to the other, and then directly behind the cell that passed to the other compartment, the tight junctions will form again, so keeping this barrier between those two compartments. Is it clear till now?
Doctor, can you repeat this idea about destruction of tight junctions?
Yes, I was saying how those cells, which are the pre-leptothene spermatocyte 1, and the primary spermatocyte that did not enter the meiosis yet, when they enter the meiosis, we said that they should pass to the other compartment, to the abdominal compartment. So how can they pass to the other compartment knowing that we have those tight junctions? They are like a barrier. So how can they pass from here to here? What happens is that when the cell will enter the meiosis, those tight junctions will be disrupted, will be hydrolyzed or broken, okay? They will be broken by enzymes produced by the Sertoli cells, okay? The Sertoli cells produce enzymes that will digest those tight junctions. In this way, they will be disrupted, and the cell that will enter the meiosis can pass from this compartment to another, so they can move forward. This primary spermatocyte can move forward in the seminiferous tubule to reach the abdominal compartment. Once it will move forward, it will move a little bit to the abdominal compartment. The tight junctions will form again behind this cell.
Okay, thank you, Doctor. Miss Al-Hal.
Okay. So they will be disrupted ahead of the cell, and then once it will move, they will be formed again behind it. Okay?
Okay, clear.
So that's what we are saying here, that the passage of the preleptotin spermatocyte from the basal compartment to the central compartment or to the abdominal compartment requires a sequential process of dissociation of tight junctions located above these cells, and then the formation of new junctions below this cell. So when the cell will move, we will have new junctions, tight junctions that will form below them. Now, what are the Sertoli cells, the functions of the Sertoli cells? So I just want to make sure that you understood that the tight junctions are only present in the basal compartment and not in the abdominal compartment. And in fact, those tight junctions, because they work as a barrier, we said, for proteins and many substances, will constitute what we will talk about now, the blood-testis barrier. So we have, they will be the main component contributing to this blood-testis barrier, because we should have a barrier between the element, and in fact those tight junctions are protecting those cells in meiosis from any protein, from any harmful proteins or elements that can come from the blood. And because we have here in the interstitial space, we have the blood capillaries, and we have migration of nutrients from those blood capillaries to the Sertoli cells, and the Sertoli cells will exchange many elements with the germinal cells. So to keep those cells in the abdominal compartment in a good state and preserve their quality, and also to protect them from the immune system, we will say why. Okay, we have those tight junctions here that work as a barrier. Okay, a barrier we call a blood-testis barrier, yani a barrier between the components coming from the blood and the lymph nodes to the seminiferous tubule. So not any molecule that can go inside of the Sertoli cell can pass through this abdominal compartment due to the presence of the tight junctions. And also those tight junctions will prevent the leaking out of the sperm cells, the formed sperm cells, when they will be released in the lumen. Okay, those tight junctions will prevent or inhibit their leaking out from the abdominal compartment to the peripheral compartment, and in this way they will protect them from any immune reaction, because the immune system, our immune system, is not tolerant to the antigens present at the surface of the sperm cells. So those sperm cells should not be in contact with the immune system, or with blood, or with the lymph vessel element of the lymph nodes. Okay, because we can have an immune response against them. That's why the tight junctions are also important. They provide an immune barrier between an immune protection for those cells, for the sperm cells. So now we will talk more in details about the function of the Sertoli cells. So those Sertoli cells, they are involved in the development and regulation of the spermatogenesis due to the factors that they will secrete and they will produce. They will produce most of the factors that are responsible for the sperm cells proliferation and their differentiation and their meiosis. And the beginning of the spermatogenesis at puberty is only possible if the Sertoli cells are well differentiated. So we should have at puberty well differentiated Sertoli cells in order for the spermatogenesis to start. If the Sertoli cells are not well differentiated, or we have a problem at the level of the Sertoli cells, the spermatogenesis cannot occur. Okay, and those Sertoli cells, they have receptors for the FSH, the follicular stimulating hormone produced by the anterior hypophysis, and they have receptors also for the testosterone. And it's under the action of those FSH and the testosterone that this they can regulate the spermatogenesis. Okay. So now those Sertoli cells, they have a support function. In fact, they support physically the germ cells inside of the seminiferous tubules by forming junctions with them. Okay. And also by modifying those junctions, like we have seen for the tight junctions, they can be disrupted and formed again in order to allow the displacement of the germinal cells. So in this way, they provide physical support for the germinal cells. They have a role in phagocytosis, as we have said earlier also, because they will phagocyte the residual bodies produced the excess of cytoplasm released during the maturation of the sperm cell. We said it will be phagocytosed by the Sertoli cells. Okay, and they have a role in spermiation. Because the Sertoli cells are attached, in fact, we said that the Sertoli cells are forming junctions with different cells of the germline. They are forming junctions also with the spermatozoa, with the formed or newly differentiated spermatozoa. And we said that the spermiation is the release of those sperm cells in the lumen of the seminiferous tubule. So this spermiation is made possible through the disruption of those junctions between the Sertoli cells and the sperm cells. So by disrupting those junctions, in this way, the Sertoli cell will be contributing to the spermiation, will have a role in the spermiation phenomenon, or in the release of the sperm cells in the lumen. So the Sertoli cells allow the detachment of the sperm cells from their apical pole, from the apical pole of the Sertoli cells, to be released in the lumen of the seminiferous tubules. They produce many factors. Also, the Sertoli cells, they secrete many factors that play a role. They have a nutritive role to the germinal cell lines. Okay, and they produce cytokines and growth factors essential for the proliferation and the differentiation of those cells. So, for example, regarding the proliferation, the Sertoli cell control the proliferation of the spermatogonia by secreting the inhibin and the activin. The inhibin, it inhibits the proliferation of the spermatogonia, while the activin produced by the Sertoli cells stimulate the proliferation of the spermatogonia. The Sertoli cells also produce the interleukin 1 and interleukin 6, which regulates the replication of the DNA. So the replication of the DNA of the spermatogonia or the germ cells is regulated by the interleukin 1 and interleukin 6 produced by the Sertoli cells. The interleukin 1 stimulates the replication of meiotic and mitotic DNA, while the interleukin 6, it inhibits this replication of the DNA. Also, the Sertoli cells produce growth factors as the insulin-like growth factor 1, IGF-1, and the transforming growth factor beta, the TGF-beta. So those are two growth factors that control the division and differentiation of the germ cells. Also, they secrete the plasminogen activator, which is an enzyme that convert the plasminogen, which is another enzyme, an inactive enzyme, the plasminogen, to its active form, the plasmin. So the Sertoli cells will produce the plasminogen activator that will cleave the plasminogen, and this plasminogen will give the plasmin. It's an active enzyme. This is a protease that will degrade the tight junctions between the Sertoli cells, and thus allowing the passage of the pre-leptothene primary spermatocyte to that started the meiosis to the abdominal compartment. And also they will secrete and also this plasmin is important for the disruption of the desmosomes and gap junctions between the Sertoli cells and germ cells. And following these disruptions of the desmosomes and the gap junctions, the Sertoli cells will be able to phagocytose the residual bodies produced during the differentiation of the spermatozoa or of the spermatids. So also they secrete transferrin and ceruloplasmin, and the transferrin and ceruloplasmin are two proteins that allow the transfer of iron and copper to the germ cells. So the transferrin transfers the iron and the ceruloplasmin transfer the copper, and in this way the germ cells can receive iron and copper from the Sertoli cells by the way of transferrin and ceruloplasmin. The Sertoli cells, they produce also the androgen binding protein, because as we said, Sertoli cells, they have receptors for testosterone, so they can respond to this testosterone. So the testosterone can enter inside the Sertoli cell, bind to its receptor and exert activities inside of the Sertoli cells. But then this testosterone can be bound to an androgen binding protein, which can transport them. This is a carrier protein, okay, which will carry or transport those testosterone or androgens from the Sertoli cells to the lumen of the seminiferous tubule. And in this way, the testosterone can be also, and it can pass in the ducts of the seminiferous tubule and in the ducts of the genital system to the epididymis and the other ducts also, the vas deferens, etc. Okay, so the ABP serves as an androgen transport protein, and it concentrates the testosterone in the seminiferous tubules and the epididymis. And in fact, it is due to, because the ABP is transporting this testosterone from the Sertoli cells to the seminiferous tubules, they will be to the lumen of the seminiferous tubules. The testosterone will be concentrated, will get to be concentrated in the lumen of the seminiferous tubules and in the epididymis. So that's why we say it will concentrate the testosterone in the lumen of those tubules. And also, as we have seen earlier in the earlier chapters, that the Sertoli cells produce the AMH, which is the anti-Mullerian hormone, and this hormone causes the regression of the Mullerian ducts. The Mullerian ducts, which if they are maintained, they will become the female, they will give the female genital tract. So the Sertoli cells produce this AMH that will induce the regression of the Mullerian ducts. Now also the Sertoli cells, they have a protection function. They protect the germinal cells. How do they protect them? It is due to this blood-testis barrier that we talked about, and most specifically, this blood-testis barrier is formed by the tight junctions, so they will protect the germinal cells by the way of the tight junctions forming the blood-testis barrier. So the blood-testis barrier are formed by tight junctions at the base of the Sertoli cells and result in the polarization of these cells. In fact, we said due to those tight junctions, we will have due to those tight junctions, not all the substances present in the basal compartment can be found in the abdominal compartment. In fact, in the basal compartment, we will find the substances directing the mitosis of the proliferative phase of the germinal cells, whereas in the abdominal compartment, those necessary for meiosis are produced. Okay. In fact, they are polarized because we said they have two different poles also, and they don't produce the same. We don't have the same production of proteins all along of those Sertoli cells. So in this basal compartment, the Sertoli cells of those Sertoli cells, they will produce factors responsible for the mitosis of the spermatogonia and the germ cells, because as you know, in this region, we have the spermatogonia, we have all the cells in mitosis until the pre-leptothene primary spermatocytes. So they are all cells that will divide by mitosis. So in this region of the Sertoli cells, we will find proteins or cytokines responsible for the mitosis. They will be secreted and will drive the mitosis of those cells. However, in the abdominal compartment here, the Sertoli cells will produce proteins or cytokines that once secreted and they will pass to the germinal cells, they will control the meiosis and the differentiation of those cells. Okay, so the Sertoli cells, they are polarized. They do not produce the same proteins in their basal pole and in their apical pole, and this difference in the profile of produced proteins and cytokines regulates the different types of divisions and different functions of the germ cells. They will regulate here in this compartment the mitosis, and in the other compartment they will lead to the meiosis and the differentiation, the spermiogenesis. And when we talk about this blood-testis barrier, we said that the major component of it is the tight junctions, but they are not the only component contributing to this blood-testis barrier. We have also the peritubular myoid cells present in the lamina propria. We said we have those myoid cells in the lamina propria. They will contribute to this blood-testis barrier. And also we have the endothelial cells surrounding the capillaries. We have the capillaries here in this interstitial space. And you know that the capillaries are lined by endothelial cells. So the endothelial cells of the capillaries, they contribute to this blood-testis barrier also. So this blood-testis barrier consists of the endothelium of blood capillaries, the peritubular cells, and the tight junctions organized between the Sertoli cells in the basal lamina of the seminiferous tubules. And this blood-testis barrier is functional only after puberty. Okay, so because before puberty we don't have all this, we don't have spermatogenesis. Okay, so it's only after puberty that we will start having this barrier, and that will be functional and will protect the germ cell line. And also, as we said, this barrier, this blood-testis barrier, controls the exchange between the seminiferous tubules and the interstitial space, and the molecules coming from the interstitial space. The barrier is permeable to small molecules, but prevents the passage of large hydrophilic molecules such as proteins. It facilitates the diffusion and allows the intratubular penetration of steroids. So for sure, this blood-testis barrier should allow the passage of testosterone and steroids in order, because the Sertoli cells, the productions of many factors that will regulate the functions of the germline is under the control of testosterone. So the testosterone should be able to penetrate inside of those Sertoli cells. Okay, so this blood-testis barrier should not inhibit the diffusion of the steroids. And also, this blood-testis barrier is an immune protective barrier for the germ cells. It will prevent any sort of contact between the spermatozoa and the immune system, because the immune system is not tolerant for the spermatozoa. This is regarding the Sertoli cells. Now we will talk about the Leydig cells. We said that the Leydig cells, yes.
sorry بس صارت 1:30 وعنا كلاس بعدي.
Okay, I will stop here. I will see if I can do a recording for the end of the day. In this way, we will not take a lot of time to finish this chapter next time. Okay.
Okay, doctor. Thank you.
Okay, thank you.